The mRNA joins to the small ribosomal unit at the 5' unstrand region. This binds to a special binding site on the small ribosomal unit. The large ribosomal subunit has a site of EPA. The large ribosomal subunit attaches to the small subunit such that the first codon is allegiant at the P binding site. A tRNA carrying the amino acid starts in the codon on the messenger RNA. Attachment of the first amino acid carrying tRNA to a binding site. A tRNA and its amino acid attaches to the A binding site. A peptide bond between the meat and an amino acid carrier at the A binding site. Our polyphenol change is now METTHR. A ribosome moves in the direction down the messenger RNA by 3 base or 1 codon switching the tRNA and change to the P binding site. The A binding site is open and is now functioning. The tRNA ejects from the E binding site and the whole process repeats itself until the stop codon encounters. The whole tRNA ejects from the E binding site and the polypeptide change is at P site. The stop codon at the A site. A release factor protein binds to the stop codon at the A binding site. Release factor proteins indicate separation of the polypeptide change, and separation translation maturing happens.
After mRNA has been transcribed, the ends of a pre-mRNA molecule is altered. A 5' cap is added for mRNA to exit the nucleus, and a 3' A tail cap is added for mRNA to stay busy during RNA splicing. Then, nucleotide segments are then removed by spliceosomes, which are made out of SNRPs. They remove introns because they are not expressed and leave exons.
A gene is made up of sequences of bases in a DNA molecule. It consists of a promoter region in the beginning and a terminator region in the end. Transcription begins when a gene Transcription of a gene is initiated when RNA polymerase attaches to a promoter region. Specific nucleotide sequences tell RNA polymerase where to begin and end. Certain proteins called transcription factors unwind the DNA strand and allow RNA polymerase to transcribe only a single strand of DNA into a stranded RNA polymer, called mRNA. In RNA, cytosine and guanine pair and adenine and uracil pair. As RNA moves along the DNA until it reaches a terminator sequence, at that point, RNA polymerase releases the RNA polymer and detaches from the DNA. Polymerase is released and DNA rewinds. It is ready to leave the nucleus and is ready to be edited